Hi, welcome back. Um, Today we're going to talk about oops, mitosis and meiosis. Um, I would like you to read this in your textbook if it's unclear so that you can get some of the details down. This is a very confusing topic. Aside from DNA and RNA, I think mitosis and meiosis is kind of foreign to us. Um, because we don't deal with it every day, even though our body's doing it constantly. so um, let's go over the PowerPoint and see how things shake out. Okay, so DNA is composed of two strands that wind around each other to form a double helix, right? These strands, what are they made up of? So what is DNA made up of? It's made up of nucleotides. They're joined together along the sides of that helix by covalent bonds. If you go back to chemistry, what are covalent bonds? Covalent bonds are very strong bonds where the electrons are shared, right? So not that you have to know this for this exam, but just so you understand, covalent bonds are really strong, right? This is what DNA looks like, sort of, completely unraveled, right? So DNA is a double helix. It's 2 nanometers in diameter, so that gives you an idea of the size. And when it's in the cell, and the cell is not going through mitosis, the DNA exists as chromatin. Okay, so number two, chromatin are kind of like beads on a string, right? So think of kind of like a little pearl necklace, right? Um, this is how DNA exists in the cell when the cell is not going through mitosis, when it's not going through cell division, because you have to have access to the DNA, because the DNA is your template for all of your instructions in your body. Okay, so the DNA exists as chromatin, or your chromosomes exist as chromatin inside your cells. And it's sort of coiled. It's not super coiled, but it's sort of coiled. Okay. When the cell gets ready to divide, the DNA goes from chromatin into chromosomes. Okay. So the bottom picture. So letter A is chromatin, letter B is chromosomes. How are chromosomes different? Chromosomes are super coiled. Okay, they're really tightly coiled. DNA. Why are they really tightly coiled? Because these guys are going to move. So you're going to take one copy of these chromosomes and stick them in one cell, and stick another copy of this chromosome in another cell. And so you don't want them slightly unraveled because you could tear it, right, and go through mutations. And that could be deadly for a person. Okay, so let me point out too that when the cell is existing as, when it's an interphase before it goes through mitosis, the DNA, exists as chromatin. This is easy to pass light through, and so you can't really see um, the chromosomes when the cell is in interphase. When the cell gets ready to divide, that's a different story. The chromosomes are supercoiled, and so you can see the nucleus because the chromosomes are supercoiled. When you pass light through these supercoiled chromosomes absorb the light, and so you see a dark color, well, dark shade. Okay, so let me see if I can move this. DNA is organized into 46 chromosomes. We also call this 23 pairs, 2N cell, or diploid. So your body cells are all, well, most of your body cells have 46 chromosomes, or 23 pairs, or 2N, or diploid. Most of your body cells are 2N, is usually what we say, or 46 chromosomes. Red blood cells, the nucleus is actually ejected, so before it's mature, so red blood cells don't have a nucleus in them, but most of your body cells have the 46 chromosomes in them. Each chromosome of those 23 pairs, or 46, consists of 2 identical copies of DNA called sister chromatids. Okay, so if you can imagine um, one chromosome has um, a chromatid from mom and a chromatid from dad. So if we go back to this picture, let's say at the bottom, letter B, this is chromosome number 1, right? One of the chromosome number 1 is called a chromatid, and then another one is called a chromatid. Okay, the top chromatid is your chromosome 1, DNA from mom. The bottom chromatid is your uh, DNA from chromosome 1 from your dad. Okay, so each chromosome that you have of those 23 pairs or those 46 chromosomes, one copy of that came from mom and one copy of that came from dad. And that gives you your inheritance. Okay, so the sister chromatids are attached to each other at a region called the centromere. So if you look at picture B, here are chromosomes, right? And they are condensed, so they are getting ready to go through mitosis. The centromere where they're attached is right in the middle. It's that middle region, okay? This is one chromosome, and one portion of that is the chromatid, and then the other portion is a chromatid as well. They're the same chromatids, they just have slightly different versions of each gene. So this would be from mom, this would be from dad. So for example, my mom had, let's say your mom has brown eyes, the code for brown eyes, and your dad has the code for blue eyes. And so you have a brown eye code and a blue eye code. They're both the same like they code for the same thing, they just have different traits or different copies of that. We call that an allele, allele. So when your cell is in interphase, when it's just doing its thing, plugging and chugging, doing its job, going through metabolism, making waste, what have you, the chromosomes exist as chromatin. Okay. Um, and so this would be letter A. When they get ready to go through mitosis, so in the late phase of interphase, they condense into chromosomes. There are two different types of division: somatic cell division or body cell division, which is called mitosis. That takes one cell, which is 2N or 46 chromosomes or 23 pairs, or diploid, and makes two daughter cells that are both 2N or genetically identical to the parent cell. What's the purpose of a somatic body cell mitosis? It is to regenerate cells. So for example, your skin cells, if you scratch your arm, you have to have a way to regenerate those skin cells, right? How do we do it? Mitosis. If you're eating Fruit Loops for breakfast and you're crunching them in your mouth and you're tearing apart some of the lining of your stratified squamous epithelium, how are you going to replace it? You're going to replace it with more squamosal cells from mitosis. Okay, and so we do cellular division, or we go through mitosis to repair cells, right, or linings. Sex cells, a little bit different. The egg and sperm actually go through meiosis. So to make the egg and sperm, we have to go through meiosis. This is taking a stem cell, like a spermatogonia or an oogonia, and taking their DNA into half of what it was. And so we're going from a 2N cell to a 1N cell. So that 1N is in the egg and 1N is in the sperm. So when the sperm fertilizes egg, we then have a 2N cell. Meiosis to make sex cells is called, cell division to make sex cells is called meiosis. Okay, so the cell life cycle. Most of the cell's life is spent in a non-dividing state called interphase. Interphase is where the cell is doing its job, right? So if it's a lung cell, it's letting CO2 and O2 through. If it's a liver cell, it is detoxifying stuff. It is growing. It is going through metabolism. It's producing waste. It's sending out signals. So this is just in a phase where the cell is living, right? And then it's going to go through mitosis if it gets a signal to do so. So there's checkpoints. And when you read the cell life cycle, we talk about gap 1 phase and then synthesis, and gap 2 and then mitosis. And so gap 1 and gap 2 and synthesis are different stages of interphase. You don't have to know that, but they're like checkpoints that the cell has to go through before it can go through cell division. Okay, so the cell life cycle consists of primarily interphase, and then there's a sliver of it that consists of mitosis. So the cell spends most of its time in interphase and then a smidgen of its time in mitosis, going through cell division. The stages of mitosis, what are they? They're prophase, metaphase, anaphase, and telophase. So when you think of the cell life cycle, I want you to think of IPMAT, right? If you're thinking of just mitosis, it's PMAT or PMAT. You don't include the interphase because interphase is not part of mitosis. Interphase is only part of the cell life cycle. The goal of mitosis is to make two genetically identical daughter cells from the parent cell. So here is a cell with its DNA, right? In interphase, the DNA is duplicated or copied, right? So you're going to make another copy of the DNA or the instruction booklet. Then through mitosis, the DNA and the copy of the DNA get split into two separate cells, right? So go to the beginning again. You have 46 chromosomes or 23 pairs. You're going to duplicate that. So you're going to have 92 chromosomes or 23 pairs plus its duplicate, or we can say 46 chromosomes plus its duplicates, or 23 pairs of chromosomes plus its duplicates, if that makes it makes more sense. So here's your 46 chromosomes plus the duplicates, okay? Then in mitosis, what ends up happening is you have the 46 chromosomes going in one cell, and then the duplicates of those 46 going in another cell, and then they split. The splitting of the plasma membrane is called cytokinesis. And so at that point, you've made two daughter cells that are genetically identical to the parent cell. Mitosis is a division of the cell nucleus. Sister chromatids are pulled apart, and one copy of each ends up in the nuclei of the two daughter cells. It has 4 stages. They break it down into 4 stages. They actually can break it down into 8 stages. So we can do, the 4 stages are prophase, metaphase, anaphase, and telophase. We could do early and late prophase, early and late metaphase, early and late anaphase, and early and late telophase. But I like to keep it simple. When are you going to use this? You have to know this process generally, because for chemotherapy or for cancer cells go through mitosis and they don't stop. So that's why they're called cancer cells. Somehow the stop code or the division code, either the stop code has been damaged or the division code has been upregulated for whatever reason, whether it's environmental factors or genetic issues or what have you. And so that can be very dangerous, right? Because then those cancer cells push all the good healthy cells out of the way. Okay, so it has 4 stages, and you would deal with this with cancer. Chemotherapy drugs target all rapidly dividing cells. Not all chemotherapy drugs, but the old ones did. And so it would, it would kill the rapidly dividing cancer cells, but it would also kill, your throat cells, because they're the digestive tract cells because they're rapidly dividing, or the hair cells, right, or the skin cells. Where else are you going to use this? I'm trying to think. Chemotherapy is a big one. Oh, genetic disorders. So if somebody has a genetic disorder, you need to be aware of that genetic disorder and how it plays out, what the different chromosomes, and we'll talk about that in a second. Okay, so mitosis, I want you to know what happens at each stage, right? So when you're doing this, when you're studying this, a good tool to study is make a chart or make boxes and fill in the boxes. So prophase, draw your picture and describe what's happening there. Metaphase, draw your picture, describe what's happening there, et cetera, and what's the purpose of mitosis and the purpose of meiosis. Okay, so let's talk about prophase. So during prophase, the chromosomes that had already replicated in interphase are going to condense, right? So what this is telling me is that when those chromosomes coil up in prophase, they're getting ready to divide. And you can actually see them under the microscope. So now that they're condensing, you can see them under the microscope. Microtubules or spindle fibers form and grow from the centrioles, right? So do you remember those two centrioles? We had one of those centrioles migrate towards the North Pole and one of those centrioles migrate towards the South Pole. And then microtubules radiate from that, and the microtubules attach to the centromeres of the chromosomes. Um, the purpose of this is that those microtubules are going to pull the chromosomes to opposite ends of the cell. And then, of course, you can't have the nucleus intact when you're going to be moving chromosomes, so the nuclear envelope breaks down. Okay, so here's a picture of early prophase. This is fluorescently labeled, so it's really pretty to look at. Here's a picture or drawing. Here are the centrioles, and then here are the spindle fibers radiating from them. So the centrioles go to opposite poles, right? The spindle fibers radiate from them. The nuclear envelope breaks down, and then the microtubules are going to attach to the centromeres of the chromosomes. So here's late prophase. Do you see how we can see these guys? If you're looking under a microscope, you can actually see them. If you're under oil immersion, you'd be able to see them better. But if we were in lab and you were looking at them in the mitosis lab that we're going to be doing next week, you would actually see the chromosomes under the microscope. And so I'll show you some pictures of those. There's some at the end of the PowerPoint. You can look up plant cells. Plant cell chromosomes are actually, they look a lot, they're easier to see under the microscope than the animal cells, than the whitefish blastula, which is uh, um, a fetal, not fetal, but it's, I can't think of the word right now. Sorry, guys. It's a whitefish embryo. There we go. So it's a fish embryo. But you can still see them nonetheless. Okay, so when these chromosomes coil up, they're easy to see, right? So you can actually see the nucleus under the microscope. So if you see the nucleus, if we're talking about lab and you're trying to identify these, if you can see the nucleus under the microscope, then you're in prophase. If you can't see the nucleus, then the cell's in interphase. It hasn't gone into prophase yet. Okay, so excuse me. Here are um, the centrioles, and the spindle fibers are attached to the centromere of the chromosomes, right? And then they're going to pull them to opposite sides. And notice how the nuclear envelope has broken down. Before they do that, though, all of the chromosomes line up on the metaphase plate or the equator. And this is the easiest stage to see. So when you are looking at slides, or when you're looking at the whitefish blastula, the metaphase plate or the equator is the easiest thing to see. And so here's what they look like stained. Here's what they look like as a picture, okay? So prophase, they condense and coil up. Metaphase, they line up on the middle. And then anaphase is the next phase. Anaphase, think of away, right? So prophase before they're supercoiling, metaphase middle, anaphase away, and then telophase the end. You can actually arrest the cells in metaphase to examine them for genetic defects. This is called karyotyping. So they'll take a cheek swab of your cells or a blood sample, and they'll stop the cells from dividing, and they'll pull out your chromosomes, and they'll line them up, and they'll match them up. So this is chromosome pair 1, chromosome pair 2, 3, 4, 5, etc. Pretty cool, huh? And then chromosome 22. These are all called autosomes because they're all the same. Chromosome pair number 23 are actually your sex chromosomes. And so pair 23 are your sex chromosomes. What does that mean? Um, it, it determines your sex. So if you have an X and an X for your chromosome pair 23, you're going to be a female. If you have an X or a Y for your chromosome 23, you're going to be a male, genetically speaking. And there are variations of this. For example, someone could have an XXY and they would they could display male and female traits. Usually they develop as a male until puberty, and then they'd have to have testosterone to keep developing as a male. If you're talking about a genetic defect in the autosomes, there's a disease where you have 3 copies of chromosome 21, and that's called trisomy 21, and that gives a person Down syndrome. So there are known genetic mutations that cause known diseases. There are different things too, like sometimes a tip of chromosome 15 can relocate over to the other one, or sometimes this can get ripped off in cell division, and so you want to be careful. There's a lot of different ways these chromosomes can be affected. Okay, so let's get back to this. So anaphase, the centromere splits, right, and the microtubules shorten, pulling each of those chromatids to opposite poles of the cell. So anaphase, here's the labeled slide, and then here's the picture. And so the chromosomes are going to get pulled to opposite sides. So this would be your 23 chromosomes, or your 46 chromosomes, excuse me, or 1/2 of your chromosomes, and then this would be the duplicates getting pulled to the other side. So anaphase are moving away from each other. In telophase, I think of telophase as an end, so it's finishing up dividing. The nuclear envelope is going to reform, right? Because you want to protect the DNA. The chromosomes uncondense, so then you have access to them again. Cytokinesis means cutting of the cytoplasm. So you're going to split the cytoplasm in half, and you form 2 daughter cells. So, and this is really cool to see. This is obvious too once you actually know what you're looking for. Here's a chromosomes, and then here's a chromosomes as well. And notice how they're at opposite poles. Okay, and then look at the cytokinesis occurring. And so this is a cleavage furrow because it's an animal cell. So this cleavage furrow is forming, and it's actually splitting the cytoplasm so that you have cytoplasm in one cell and cytoplasm in another cell. And then the nuclear envelope is going to reform around the chromosomes. And so here's a picture. Nuclear envelope forms around the chromosomes. Here's a cleavage furrow splitting the cells into 2, so splitting one cell into 2. And then these chromosomes are going to uncoil. And that's telophase and cytokinesis. I consider cytokinesis part of telophase. Stop signals. So what regulates cell division? So when the cells get too big, so when they contact each other, then they stop. You have growth inhibiting factors produced by repressor genes. Sometimes environmental, like environmental pollution or like epigenetics, you know, whether it's pesticides or what have you, they can inhibit these growth inhibiting factors so that the cells stay on and they keep going through mitosis. UV light can damage this, right? And so that would give you skin cancer. Telomeres are the little end caps of the chromosomes, and these guys get worn out, and that causes aging of your cells. And so take a look at the picture. The chromosomes are blue, and then the telomeres, little end caps, are yellow. As these guys go through mitosis, you know, I don't know, let's say 100 times a day, These telomeres, these yellow regions, get smaller and smaller. So they get ripped off a little bit or spliced off a little bit as these guys go through cell division, and this causes aging. And so that is the aging process in people. Let's see. So the cell cycle includes interphase, right? Plus prophase, metaphase, anaphase, and telophase. Cell division or mitosis only includes PMAT. So cell division or mitosis is just your prophase, metaphase, anaphase, and telophase. So if you think of this, it should make sense because the cell life cycle is interphase plus cell division. And then cell division, which is also called mitosis, only involves the steps of mitosis. So let's take a look. These cells are in interphase. So these are whitefish blastula cells. So they took a whitefish blastula or an embryonic whitefish, and the ball of cells, it's just a rapidly dividing ball of cells. And so they took cross sections of it and laid them out on the slide. And so if you are interested in seeing more of them, you can just look up whitefish blastula slides and you'll see a bunch of them. And you can use Google Images or what have you to click through them and identify the different stages. I think that's the easiest way to understand what's going on. You'll have to identify these in the lab midterm exam. You won't have to identify pictures for the lecture exam, but you'll have to know the different stages and what they represent, like the definitions. Okay, so here's interphase. So I can tell these cells are in interphase because you can't really see the nucleus, right? So here's the nucleus, here's the nuclear envelope, and then here is the plasma membrane. Okay, so this is one cell, here's another cell, and here's another cell. These dots that you see, the black dots, those are actually fat droplets. So those don't count. Prophase, look at how those chromosomes have condensed. Really cool, right? And then you see the centrioles are close to that, and then the microtubules are radiating from the centrioles, and then you can tell the nuclear envelope has degraded because these guys are starting to spread out and line up. In metaphase, they look like stitches. And so metaphase, the chromosomes and their duplicates are all lined up on a central axis. This is the easiest one to spot in a microscope. Anaphase, the chromosomal pairs are moving away. Now, anaphase is a really hard one to see under the microscope because the cells don't stay in anaphase for very long. And so if you're looking for anaphase, this still kind of looks like metaphase to me, but if it's anaphase, there's going to be a little bit of space between the pairs. So here will be the 46 chromosomes and then the duplicates, and then you'll see a little bit of space in between there. Here's early telophase, so this would be telophase or telophase, however you want to say it. Notice how the centriole is in this region. You have the microtubules radiating out everywhere, and then you have the chromosomes. So the chromosomes have moved to the side. So there's your 46 chromosomes, and then here's your 46 duplicates stuck to the side. Late telophase, the plasma membrane is going through cytokinesis. So you're cutting one plasma membrane into two. And then notice how the nuclear envelopes are going to start reforming. And you can already tell that the chromosomes have started to uncoil in this one because they're not as visible. In this one, they still look like they're super coiled, right? So this is the last, this would be cytokinesis for telophase. All right, let's see. So if you can't see the nucleus, like right here, it's an interphase. So this one would be the interphase, that one would be an interphase, 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 okay? If you can see the nucleus, then I consider that to be in prophase. So I know this is labeled interphase, cross it out. Actually, is it labeled prophase? I can't read. No, it's labeled interphase. So this would be prophase, right? So if you can see the nucleus, if there's a dark nucleus inside the cell, that would be prophase. This one is metaphase because you can see them all lined up in a straight line. This one would be telophase. You can see them sort of connected. Here's one group of chromosomes, here's the other one, and then they're going through cytokinesis. This is a better picture. So what stage is this cell in? The chromosomes are all lined up on a central axis, so that would be metaphase, right? What about this one? Here's a cell that's almost split into two. That would be telophase, good. What about this one? You can see the chromosomes. So that would most likely be prophase. It's hard to tell. It kind of looks like you're looking at a weird angle. This one, you can see the chromosomes are all in the central area. That would be prophase as well. What about this one? The chromosomes are on opposite ends, so I would call that telophase. And what about this one? Telophase. Good. Okay. So let's talk about meiosis. So moving forward, or actually, sorry, let me go back. For lab, so for the white blood cell portion of the lab, for this coming up lab, what I want you guys to do is I want you to look through slides and draw pictures of the cells and the different stages of um, mitosis. Okay. You should be able to identify them. How do you identify them? I would just search whitefish blastula on Google or Bing and do images and click through them and just quiz yourself on them. That's the easiest way to do it is you look at about 100 of them and then you start figuring stuff out. All right, meiosis is what sex cells do, okay, or to make sex cells. So cells go through meiosis 1 and meiosis 2. So they have two uh, division cycles. So you can think of the egg and sperm, or the stem cells, they go through meiosis 1. It's kind of like meiosis, and then they go through meiosis 2, which is like meiosis again. It results in 23 single chromosomes, not 23 pairs, excuse me, like in mitosis. So you have this stem cell, which constantly kicks out a pre-sperm cell or a pre-egg cell. And then the pre-sperm and pre-egg cell go through meiosis 1 and meiosis 2. The result of this is 23 single chromosomes. We call this one end or haploid, not 23 pairs, right? This haploid number is important because when the egg, which has 23 single chromosomes, and the sperm, which has 23 single chromosomes, fuse, you have 23 pairs or 46 chromosomes or 2N, right? So the egg and sperm have to have half of the genetic information so that when they join, you have a full complement of the total genetic information. If they didn't go through this reduction phase, if they didn't go through meiosis, they just went through mitosis, you'd have way too much DNA, and that fertilized egg, that zygote, would die. Okay, so on the left side is mitosis, right? Here's your parent cell. The DNA duplicates, so you have 46 chromosomes and their duplicates, and then it splits into two daughter cells. So here's your 46 chromosomes, here's your duplicates, your 46 duplicates. Done. Okay, meiosis is a little bit more complicated. So here is the parent cell. So this would be a pre-sperm. It's actually, you have stem cells in your, well, in ovaries and testes, and they divide and kick out these like pre-sperm or pre-eggs, if you will. And then these guys are going to go through meiosis to make the egg and sperm. So here's your cell. The DNA is going to duplicate, right? And then the DNA splits into two daughter cells, just like mitosis. It's a little bit different. So you get mixing of genes and whatnot. That's why if you have siblings, you don't look exactly like your siblings. So there's some crossover of genes from the same chromosomes. So you have 46 chromosomes in this one, and then 46 chromosomes in this one. Okay, that is called meiosis 1. And then meiosis 2 is splitting that information into half. So 23 of those chromosomes go here, and then 23 of those chromosomes go here, and 23 and 23. So you have half the genetic information in those chromosomes or in those cells. And so the parent cell was 46 chromosomes, and each daughter cell you have 23 single chromosomes. So from one uh, sperm cell or egg cell, how many eggs or sperm do you make? Good. So you make 4 sperm, right? So one sperm stem cell, excuse me, one sperm cell, you're going to make 4 sperm. From one egg cell, you're going to make 4 eggs. And it actually doesn't quite work that way. Um, whoops. Because 3 of the eggs are reabsorbed and 1 is functional. All right, so here is what it looks like. I'm not going to ask you this in lab. I want you to know it for lecture, though, like what happens at what stage, but you don't have to identify it through the microscope in lab. Okay, so meiosis 1 is separating the homologs. Prophase 1 is these guys condense, and it says 1 pair of homologs or chromosomes, homologous chromosomes, and then another pair of homologous chromosomes. And then you get crossing over of gene traits. Okay. Metaphase 1, these guys align. So your homologs align, right? And notice how, do you see this pair in the box? How this one has one of the segments of chromosome from this one, and this one has one of the segments of chromosomes from that one. This is called crossing over. And that's why you have the same codes for stuff, but that's why you and your siblings don't look the same, because you have actual different traits that, like let's say this is a code for hair color, but this one represents red and this one is non-red. And so you got the red one, your sibling got the non-red one. That's going to give you guys different hair colors. Okay. So metaphase 1 is they're lining up on a central axis. Anaphase 1 is they're moving away. And then telophase 1 is like they've separated cytokinesis. But they're not going to stop there. They're going to go on to meiosis 2. So meiosis 2 is, and notice how it says there's no DNA replication. Meiosis 2 is just reduction. And so you're going to take these chromosomes, these 46 chromosomes, and they're duplicates, and then you're going to split them into ultimately 23 chromosomes, 23 chromosomes, 23 of the duplicates, 23 of the duplicates. So this is prophase 1, or excuse me, prophase 2. Metaphase 2, they land up. Anaphase 2, they move away, and telophase 2 throughout the end. And so from one cell, we back up, we got how many egg cells or how many sperm cells? 1, 2, 3, 4. Okay, this was 2N or 46 chromosomes or 23 pairs. These guys are 1N or haploid or 23 single chromosomes. Okay, that's it, you guys. Good job.